Hello, my name is Teresa Snyder-Leiby. Welcome to today's GeneMarker HID software for paternity testing webinar. GeneMarker HID software is an expert system for forensic STR DNA analysis. The basic functions of GeneMarker HID are covered in the introductory webinar. Please review the introductory webinar prior to beginning specialized applications. In today's webinar, we will briefly review the basic GeneMarker HID functions before using a demonstration set of data in the paternity testing applications of GeneMarker HID. These include automated pedigree drawing and paternity index calculations. The program uses the equations from the Association of American Blood Banking, AABB, the standards for relationship testing laboratories for trio PI calculations and motherless case PI calculations. The program provides immediate visualization of paternity exclusion or inclusion, displaying allele conflicts in both pedigree diagrams and sample traces. Additional webinars for relationship testing in GeneMarker HID include database searching for reference samples or familial search with likelihood ratios from identity by descent calculations, which are helpful in cases such as missing persons, unidentified human remains, and paternity. There is also kinship analysis, which uses identity by descent calculations across 3 generations. As a review, GeneMarker HID is an expert system for human identification genotyping. GeneMarker HID is compatible with all CE instruments and human identity chemistries for rapid, accurate size and allele calls. The program contains user-friendly linked navigation, which decreases analyst strain and saves time. It contains automated control and allelic ladder concordance checks and the ability to customize parameters for each locus, including the analytical range, stochastic range, and stutter filters. The user management provides control of access rights and an audit trail. GeneMarker HID has many different reporting options, including print reports, tables, and CODIS formats. As a brief review from the introductory GeneMarker HID webinar, GeneMarker HID accepts raw data files from all major capillary electrophoresis instruments, including FSA, HID, RSD, ESD, SCF, and SG1 files. The user management allows the administrator to assign access rights and password access to each authorized user and provides a complete audit trail. The Run Wizard template simplifies analysis. Please review the introductory GeneMarker HID webinar for details about creating and saving Run Wizard templates. Confirm the settings in the second screen of the Run Wizard and click Next. In the final Run Wizard screen, select OK to apply the parameters. Analysis is rapid, taking approximately 20 seconds to analyze 96 samples. The file name tree at the left has 2 quality indicators. Red question marks alert the analyst that these samples have caused one or more of the analysis rules to be fired and will require review. The green sheet in front of each of these file names indicates that the size calls were all of high quality. We see a summary of the analysis at the bottom of the screen. Icons of common interest include the trio of Show Hide toggles to remove the gel image, name tree, or report table. Use the multicolored icon at the top of the screen to toggle to the single die view of each trace. Please refer to the introductory GeneMarker HID webinar for details on report table format options and size calibration verification. In this view, the synthetic gel image is turned off. The report table is in vertical form and the FAM trace is selected. The latter error reported can easily be seen by zooming in. Draw a box from the upper left to the lower right of the area of interest. The color coding for the latter error alerts the analyst and indicates that the peaks are in the expected bins, but that 2 of these virtual alleles have higher than expected RFU. Yellow flagging in the report table to the right indicates that one or more of the analysis rules were fired for these peaks. Linked navigation in GeneMarker HID provides rapid access to these areas of the electropherogram. Simply use a double left mouse click in the yellow flagged cell of the report table to zoom in on this sample and locus. By using the peak table icon, we may toggle to see that the rule that was fired was heterozygous imbalance. The help in the upper right menu contains a list of quality flagging abbreviations. Many labs prefer to view results in the all-color display. Please refer to the introductory GeneMarker HID webinar for details on these features. GeneMarker HID has flexible reporting options. In this print report example, we have the files grouped by dye for the alleged father, child, and mother samples. Use the Applications drop-down menu to activate the Relationship Testing application for automated pedigree diagrams and paternity index calculations. Select the Sample tab at the top of the table to display all of the files from the project. In this project, we named the files with a number for the family identifier separated by underscores from the relationship level identifier. By selecting the Tools Family Group tool, we will use this information to group the files by family. In the lower right side of the screen, set name identifiers for child, alleged father, and mother. At the lower left side of the screen, group the files by sections, mixed position, or order, then click the Match rectangle. The files for each family are grouped together. Click on the OK rectangle to automatically draw the pedigrees. Using the Family drop-down menu, select each family's pedigree. In this motherless case, we have 3 children. The second child has a single allele conflict with the father at locus FGA. The allele conflicts are noted with a red node for the child, and the allele call is in red font. The linked trace provides a visual of the data at this locus. Use the Family drop-down menu in the center of the toolbar to display the pedigree diagram. Selected files are numbered in the selected column of the file list on the right. This pedigree is an example of paternity non-exclusion. There are no allele conflicts between the parents and the child. The genotype of each individual is displayed within the pedigree diagram. If there were any allele conflicts, the allele calls would be in red font. Right-click on the diagram node of the child, in this case a circle indicating female, to activate the pop-up menu. Click on Select Family to display the electropherograms of all of the individuals. Use the color icon at the upper left to toggle to each dye channel to review the traces for the family. To calculate the paternity index, right-click on the node of the alleged father. Use the pop-up menu and select Enter Node to add individual information, and select Contested to activate the paternity index calculations. Select the appropriate allele frequency for calculations using Tools Allele Frequency in the toolbar. The program has several allele frequency tables for major U.S. populations, including the CODIS 13, Identifiler, PowerPlex 16, and ESI loci. Allele frequencies from other populations may be easily imported using the open folder icons. Details for formatting these tables are provided in the linked user manual. The paternity index is displayed in the PI cell for the sample. Select the Calculation Detail tab to display the results for every locus. Use a right mouse click to copy and paste the table into an existing report, or save the table as a text tab delimited file. This is an example of exclusion of the alleged father. The allele calls that are in conflict with the parent are in red font in the pedigree. The node of the child is outlined in red. When the mouse is held over the node, the alleles that are in conflict are displayed in a drop-down list. Click on each marker to display the 3 electropherograms. Some of these may be uninformative, as with the D8S1179. Others, such as CSF1PO and D5S818, indicate allele conflict with the alleged father. Allele calls that are consistent with uniparental disomy are displayed on the same side of the pedigree diagram, as is the case with FGA in this example. In this motherless case, we see exclusion of the alleged father at all of the loci with red font in the left paternal column. D21S11 and CSF1PO allele conflicts are displayed here in the trace view at the right. In this example of inclusion of the contested father in a motherless case, the calculation details are displayed using the motherless case equations and may be copied, pasted, or exported using a right mouse click in the table. Save the pedigree drawing for each family using a right mouse click anywhere in the gray area around the pedigree and select Save Export Image from the pop-up menu. Save the pedigree file using the Save icon in the toolbar. The file may be reopened at a later date and additional children can be added to families. If you prefer to draw the pedigrees, select the file New Pedigree. Then fill in the identifiers and select the genotype from the Current Project drop-down menu or from the Relationship Testing database. Right-click on the node and select Add Mate. Provide the identifiers and genotype, then right-click and select Add Child and repeat the process. Select the allele frequency and display the PI results using the same steps as covered in the previous slides. Thank you for participating in this GeneMarker HID software paternity testing webinar. The program has automated pedigree drawing and paternity index calculations using equations from AABB Standards for Relationship Testing Laboratories. These include trio PI calculations and motherless case PI calculations. The program has immediate visualization of paternity exclusion, displaying allele conflicts in the pedigree and in the sample traces. GeneMarker HID is an analyst-friendly program which has been validated and concordant in many laboratories. The program has accurate, rapid analysis of forensic STR data, including quality flagging, size calibration checks, and automated control concordance. It is compatible with all major file types and human identity PCR kits. The user management provides access rights, control, and audit trail. The report flexibility includes allele reports, peak tables, and CODIS formats. Thank you for reviewing this webinar on paternity testing with GeneMarker HID human identity software. For more information or to download a free 30-day trial, go to www.softgenetics.com or send us an email at info@softgenetics.com.